We are back. We are back. Okay. I think it's because the weather is slowly progressing to worse. Yeah, the weather is getting. It might start storming in a minute. So let's get through this paper as quickly as possible. Two-way ANOVA. The reason why is because if you look, it's cross time, so they have to do ANOVA. Hey Tapper, how's it going? Hi. The weather is just horrible. How's it going? We're looking at papers today, right now at least. I'll be doing something else after this. It's just I felt like I want to start my days with. Nah. I think all the internets are cursed. I think it's all cursed right now. But yeah, two-way ANOVA. So I think the reason so they did a two-way ANOVA largely because it is a temporal data. So they're doing an ANOVA for the temporally, but they're also doing a two-key Kramer because they only have two conditions. So that's kind of the way to look at it. All right, glad it's working. All right, so here we go. So figure 5 is abrasions of FAO by of the fatty acid oxidation by deletion of this protects from IR damage and enables heart regeneration. So then I think figure 5. Well, this is a very long paper. Oh, how many figures is this? 5 extended. Not. Oh, yeah. They have two. They have a lot of extended figure. They're making a very bold claim in this paper. A very bold claim, and so I can kind of see why they have all the data. Figure 3. I think maybe we should skip some of the extended figures. Yeah, the extended figures are supplementary. So let's just move on to figure 3. So this is impeded mitochondrial import of fatty acid rewires the metabolism of cardiomyocytes and boosts AKG levels. If we remember, AKG was what is involved in the in regeneration, pretty much. And if we look at our nice figure right here, actually, you know what? I'm gonna just zoom in a bit, like so. Let's zoom in this way. Let's zoom in this way. All right, so we have OCRs. What does OCR stand for? Oxygen consumption rates. All right. So the oxygen consumption rate in the heart after the BSA. Yeah. Oh. Crazy. So let's see here. So, and they're using LCFA, BSA. So long chain fatty acids, and then medium chain fatty acids, and then, oh, I wonder where the bottom, oh, all right. I know someone showed up and I ignored them because they were a bot. If you're talking to them, just ignore them. Let's see, short chain fatty acids using the Mito Stress Kit of Seahorse Instruments. All right, so LCFA and MCFA, long chain and short chain fatty acids. So here they're looking at the BSA and then the BSA of the long form, and then some more BSA and then BSA of the long form. Okay, this is very much out of my forte. I wonder what this is. I wonder what this is referring to. This is oxygen consumption rates. I think you have to know what the model or the methodology to explain for this. So, if anything, we need to go to. Oh, this is another extended figure, isn't it? Oh, too many extended figures. Let's just look at this one for now. In fact, we'll just go ahead and load it up in another. We'll just load it up in like we've been doing. Right here. All right, let's see here. How are things faring here? Oh, look, Call of the Abyss. Decanal COA. All right, so we have the control, knockout. We see a decrease in these. They're significantly decreasing in fold changes. We'll have to see what this stands for. Here's more gene activations. Oh, that is huge. Yeah, I think the gist of it. I think the gist of some of these figures, they kind of showed the main point here. We're getting heart regeneration, and now they're validating it. Here, they're going through all the genes to show off which genes are involved. And they're also showing off, so here you can see like the different genes that are being upregulated, which are involved in regeneration. So alpha KG was one of those said genes, and it's significantly upregulated. Then here we can also see, if we head on down, here are even more treatments and more genes. So fold changes in diglycoglycerols, triglycoglycerols, the COAs, in CPT1B knockouts. So CM specific inactivation of OGL-DH in adult mice induces hyperplastic and hypertrophic cardiac growth, similar to the CPT1B deficient mice. So now they're elucidating the pathway. Ooh, interesting. They're now elucidating out their pathway. And here they ended up creating a similar mouse. This is, again, they're using the Cre recombinase to knock out OGDH. I think this is one of the genes that they identified up here, isn't it? I don't see where they reference the gene up here, but interesting. So they knocked out this one, and then they end up getting similar results. And this is quite a significant fold. Wow, some of these are huge. Look at the fold change on some of these boys. So yeah, pretty much they here you can see controls, knockouts, the heart, similar. Wow, actually a lot of similar stuff going on when it comes to the heart. Oh, a lot of similar stuff. So OGDH is most likely involved as well. I wonder if there's I wonder if they try and show a connection between the two. And this is more sequencing. Let's see here. Oh, there's positive cell deregulation again for their knockout. Let's see here. I'm just kind of skimming through because this is all genetic data now to kind of highlight what's the differences they're coming up with, what they're seeing. The analysis of the epigenetic and expression changes in the cardiomyocytes after inactivation of these two genes in adult mice. So again, this is all quantifications, and they're showing the differences that are going on. And there's actually quite a good number of differences that one can see. And then accumulation of alpha KG stimulates KDM5 activity, attenuates maturation, and enhances proliferation of cardiomyocytes. Ooh. Oh yeah. So let's go into this figure and start kind of breaking it down. See exactly what we want to look at. And here we'll load this up. Let's have a peek at you. Yeah, PH3 and Ki67, which are both proliferation markers. So we do see an increase in the proliferation. We see an increase in the KG, alpha KG. We see the processes that are being downregulated and upregulated. Oh. Vasculogenesis and protein phosphorylation. Vasculogenesis, which means we have a decrease in the vasculature formation. All right. I wonder why that's being downregulated. I can understand protein phosphorylation because when you're doing cell cycle, the last thing you want is start adding phosphorus to all of your proteins and inactivating them. So yeah, I can understand that one. And cell differentiation is also downregulated, though. Cell DNA repair, cell cycle, cell division. So all of the cell proliferation markers are up. But that is down. C. Let's get over the C real quick. I want to know something. Where are you, C? Here you go. Enrichment of, let's see, expression of the genes of DMS and KG treated neonatal cardiomyocytes. So the alpha KG treated neonatal cardiomyocytes. DMSO is just it's dimethyl sulfoxide, as you can see right here, and it's just a saline that people use as a control. So yeah, it's a basic control. But I'm in. So yeah, if I remember, this is involved in proliferation. So now the big question is, I have questions. So this is the single time point. I would love to know when this time point is. It's probably, if I look up figure 5 I'll probably find that out. Let's look it up real quick. Fig. Dot 5. Okay. Figure. All right. I wonder where figure 5 is. In here. All right, I guess. Oh, here we go. Oh, I get it. All right, right here. Found it. Okay. So to confirm the accumulation of alpha KG, the critical signal to prevent the maturation and enable proliferation, we treated there it is right here. Key 01 to 1 neonatal cardiomyocytes for 4 days with cell permeable alpha KG. We observed a marked increase in Ki67 and PH3 and CMs, consistent with RNA-seq data, indicating enrichment of the Go terms related to the cell division in AKG-treated cells. Furthermore, the level of expression of NPAA, NBB, actin, and MH7 genes were predominantly expressed during fetal and neonatal stages, was strongly elevated, whereas the level of expression of genes associated with CM maturation and differentiation include TNI3, MILK3, and MyoCD. Now, these were reduced. Treatment with alpha KG also diminished the H3K4M. We get a funny name for some of these instead of these long. All right. Anyway. Let's see here. Do not affect these, and in line with the findings of our expression of enzymes and CMs that increase the levels. So pretty much figure 5, there is no treatment. There is a treatment here, but the idea is that they wanted to see if there was any potential effects involving, or what happens if you just upregulate alpha KG? Do we end up getting something similar involved when it comes to development? It's another form of control. Pretty much, they're trying to show that it enhances proliferation in general. It's not because of damage. There's a major there is a major okay. There's a difference between there's multiple differences when it comes to different pathways. When it comes to proliferation and will you stop that? All right. So when it comes to development and regeneration, these are two different cycles, two different stages. Development is as you start from something that needs to develop into something else. While regeneration is you had damage, something that was lost, and it has to grow back. So the environments are completely different. And as such, the gene expressions will be different here and there. Likewise, there's a difference between proliferation during development and proliferation during regeneration. Because in development, you have stem cells which will start differentiating, and then once they proliferate, and then they differentiate to the specific cell types. While in regeneration, you will have de-differentiation, where you'll have these highly specialized cells becoming stem cells, then they will proliferate and then differentiate. The pathways will be the same or similar, but not identical. So what they're trying to show here is that with alpha Kg, it will just naturally promote proliferation regardless of when it is being utilized, when it is being upregulated. It's not just a damage thing. It happens even during development. If that's from, at least this is what I believe, this is my understanding, because this is all about, these cells, cultured cells, and the expressions that are going on, following treatment with alpha Kg. So yeah, alpha Kg is upregulated. Alpha Kg is upregulated. We also end up getting when you get upregulation, you end up getting upregulation of a bunch of other these. Oh, okay. So we are reviewing this paper here for fun. It is called Inhibition of Fatty Acid Oxidation Enables Heart Regeneration in Mice. For a quick recap, because it has been an hour and 40 minutes. So a quick recap. In mammals, when you have heart damage, you do not recover from the heart damage. It is, when you have heart damage, you just don't recover from that. And because of that, there is a major push in science to find ways to recover from these types of damage that are irreversible. And here they found this paper, which was published in Nature last year. So this is a high-impact paper, high-impact journal. This paper published saying that in an organism that should not be able to regrow the heart, when you inhibit oxidation of fatty acids, you actually can have heart regeneration, which is kind of almost unheard of, especially in a mammalian model. Zebrafish can do it all the time. In fact, if you just go to Google, or actually I'm using DuckDuckGo, but if you go in heart regeneration, if you go in here, type in heart regeneration, I think the very first thing that you'll come across would be a zebrafish. Hey, right here, look at this, zebrafish. Fish are notorious for regenerating the heart. Same with axolotls and salamanders. They're really, really good at it. Humans and mice and all other mammals, not so much. So yeah, this is very, very important. This is very, very intriguing. So we're going through right now, looking at the figures and looking at the data, trying to figure out if what they're, do the figures kind of support their claim, and what kind of information can we glean out of it. And so far, there's a whole lot of stuff. For instance, apparently the fatty acid oxidation is caused by a gene called CPT1B, or at least it's involved. CPT1B is involved, and by knocking it out using a particular transgenic model, you can actually end up having an increase in proliferation. So this is this data here, showing an increase in, this is tunnel, this is cell death here. Where are you, proliferation? Here, increase in proliferation. They're using, that's tunnel as well. Yeah, where are you? Are you here? Here it is, right here. Ki67 and pH3, here we go. Proliferation. So you get increases in proliferation of in these cells when you end up having, when you knock out CPT1B. And one of the genes involved in proliferation is alpha Kg, which is one reason why we started looking at the next figure here. Yes, I usually don't. I don't. Yeah, I wanted to talk science today, so I figured I'm going to have fun talking science. But yeah, so because of this, they ended up showing off in this paper, a lot of in this paper that's, I mean, let's see, where are we right now? We're on figure 5. Was it another extended figure? Oh, the discussion. Okay. Um, Here it is, figure 5. So yeah, pretty much they want to, when it comes to these types of papers, whenever you get into a higher impact paper, you kind of have to come in at all angles because there will be lots of questions asked and a lot of questions that will be brought up. For instance, alpha Kg, it is upregulated in your model. Is it because of the damage, or is it just because of, is it because of your inducing damage, or is it because of the fact that, it is alpha Kg? In fact, if you upregulate it, if you're trying to build a genetic pathway, which is exactly what they're trying to do in this paper, does alpha Kg, is it just a correlation that you get proliferation when you end up upregulating alpha Kg, or is it some other factor involved, and this might be involved somewhere else? I mean, we kind of already know what alpha Kg does because of the fact that there have been many other papers in the industry which show it off. But one thing about science and one thing about research is that it is very redundant. Just because we know alpha Kg is involved in proliferation doesn't mean it might be involved in proliferation here. You have to prove it regardless. So this is what figure 5 is kind of showing off. In their model, and when they upregulate the alpha Kg, actually I would say regardless of it, alpha K. Okay, let's go find. Let me go get up here and show you alpha Kg. All right, we need to go back up here. Yeah, this is why I hate acronyms. This is why I hate acronyms, because as soon as you start talking acronyms, which is something that happens very frequently, put that? When you start mentioning acronyms, you quickly run into the issue where everything, people get lost. Here we go. Alpha ketoglutarate. So alpha ketoglutarate. And if we go over, let's go over here to look up alpha ketoglutarate. Alpha ketoglutarate. Right here. It's a biologically important compound. It is produced by delamination of glutamate and is an intermediate in the Krebs cycle. The term ketoglutaric acid, when no further qualified, almost always refers to alpha. So here we can see that it's involved in the Krebs cycle. Alpha ketoglutarate is a key intermediate of the Krebs cycle, and it is after isocitrate and before succyl CoA, which is the other part of the Krebs cycle, which we were seeing in the figures. It also happens to be a nitrogen transporter. It's involved in the formations of glutamates. It is mediation of molecular recovery. It's an antioxidant. It apparently, studies have linked it to an increase in lifespan of nematodes and worms. It has been showed to be that in glutamine-deprived conditions, the alpha ketoglutarate promotes native CD4 T cell differentiation, so it's an immune regulator. It's an enzyme cofactor. You can kind of, it's involved everywhere. It's kind of involved everywhere. And I bet if we type this in with, because I remember this being involved quite a bit in regeneration. See right here, complex as a hub for plasticity in regeneration. So it has been, and here's our paper we're currently looking at right here. So it's involved in the Krebs cycle. It's involved in the, involved in energy production, but it also happens to be associated with many other aspects. One of them happens to be regeneration. And one of the, if you remember when we looked at it here, back at, back here in our wiki article, it is a major player. Sorry, where are you? It's a major player in the formation of amino acids. When you're regenerating, you need to do cell proliferation, which requires DNA duplication. And DNA duplication requires amino acids, which means this is needed. So this is one reason why I comment saying that it's important in regeneration. All right, so here we are. Okay. So they're showing here that it increases in alpha ketoglutarate, which is involved in Krebs cycle. Yeah. Hey, my internet dropped for a second. It dropped for a second. It dropped for a minute. But yeah. So this Krebs cycle upregulation, and we end up seeing that when you treat developing organisms with this, you get an increase in proliferation and a decrease in cell differentiation, as well as an upregulation of many other hallmarks involved in, DNA repair, cell cycle. The fact that we actually, let's hop back over to this figure real quick so I can kind of zoom in. The fact that we know that alpha Kg is involved in, amino acid synthesis, it kind of really it's pretty obvious it's involved in cell cycle, DNA repair, because you're repairing the glutamate, the glutamines. Your DNA repair, which means you're going to be replacing damaged amino acids, or you're going to cell cycle, cell proliferation, or the duplication of the DNA. I just forgot what the name is of it called. Oh, mitosis. All right, mitosis. So yeah, it's expected. This upregulated would see increases in here. And then likewise, because differentiation is involved in, something is involved in specialization of these cell types, then of course we're going to see decreases. We still have to prove it in papers. I mean, we still have to show this off because, not everything that's involved in proliferation is connected to this. But nonetheless, it's fun to see this type of cross-validation going on. And it's fun to see all of the... No, that's fine. That's fine. I highly doubt a lot of the people who originally showed up. I did have someone show up on YouTube because I'm streaming it on YouTube as well. I'm streaming it over on the lecture hall channel because this is a paper. And let's see here. So yeah, so it's fun to see to see this kind of confirmation. And then I'm guessing figure 6. Yeah, this is an extended figure. So this is pretty much validating what we saw in figure 5. And I bet if we look here in the literature, yeah, figure 5 and figure 5 and then extended figure 9. Yeah, they're associated with each other. So yeah, this right here is confirming the data here. Oh, ask questions away. I'll answer whatever you want. Ask questions. That is how one learns, is by asking questions. I will gladly answer questions. I think one thing that is often forgotten is when it comes to learning is I would actually make an argument. It's because of the fact that in academia, and particularly in high school, middle school, primary school, like they tell you be quiet, don't ask questions, just listen to the teacher, which is not the proper way to learn. It is never the proper way to learn. There are proper ways to teach, and one of them is to always get people to ask questions. Asking questions is the way to go. And sadly, by the time people make it to college and undergraduate and then eventually graduate school, they forget how to ask questions, and as such, they go through never really learning never really learning how to ask questions, or never really learning the process to ask questions. And as such, so yeah, ask away. You have a question? Ask away. Don't know what this is? We'll go through it. All right, let's see. And this should be figure 10, which should be finally finishing off the paper. I believe figure 10, yeah, right here. Let's see here. Oh, extended 10 is... This is what you call science. Well, yes, this is science. But it doesn't have... It's called the scientific method for a reason. It's... You ask questions, you revise your hypothesis, and then you ask more questions. It is the... Oh, no. It just takes time to understand it. All right, it just takes time to understand it. Where would you like to... I wonder if that would work. Oh, it did work. Okay. Where would you like me to position so that I can try and help you understand? Yeah, the annoying figure. Can I zoom in myself? Oh, I can. Okay. All right, then. All right. So while you are on your way, what I'm going to do is finish the paper off. So here we have the z-scores, looking at more genes and their fluctuations. Let's see, it's a heat map of gene expression of the KDM5 family members at different stages of heart development and after transaortic constriction. So they're looking at development and then after. So, oh, interesting. So I mentioned it earlier. I mentioned it earlier. There is a difference between development and a difference between the regeneration pathway. And it is important to understand them because you don't want to reactivate development. You want to activate the regeneration pathway. So here what they're doing is they're performing a heat map during the development and the damage to try and identify exactly. They're trying to identify exactly where which genes are responsible. It might be all of them. It might not. It might be none of them. Let's take a quick peek at this basic heat map. Let's see here. Let's take a look at this basic heat map and have a peek. Here's our heat map. So KDM5A, B, C, why? Ooh, interesting. There are so this is there's 4 genes all called KDM5. And then here are all of the different stages. So actually this is kind of interesting. There's a very intriguing, very intriguing. So to explain, this is upregulation, downregulation. They used 1 to -1. You see here that's so the richer the color, the darker the color, the more closer to 1, the more lighter, the closer to 0, and the darker blue means the more to -1. And then here you can see E14 to neonatal 2. So these 4 are all development. Adult 1, adult 2, and then the constrictions here, these are all, these are all fully grown or being treated. So if you look, just basically, the KDM5 family is involved. Okay, the first A, B, and C are involved in development, but they're not involved in... they're involved in development, but they're not involved in adulthood or damage. However, KDM5D is not involved in development, but it is involved in their model or in the damage. That's D. So let's see here. There's our control. Here's our KDM5. But why are they looking at B of all of a sudden? Let's see here. There's B, 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 B. More Bs, more Bs. More Bs, more Bs. KM5 more Bs, and then this is the final thing. So why did they isolate out B of all of them? When you look at A, B, and C, there's almost no difference between the three. Is it because of the fact that neonatal 2, there's a large increase here? I mean, that might be why. Let's check out the discussion and see. So we're looking for 10A. Here we go, 10A. KMB plays important roles during heart development and expression progressively declines during CM maturation. KM5B knocked. Well, yeah, we know this from a paper that was published in 2013. This hypothesis, let's see here. Pharmacological inhibition of alpha-KG dependent effects by the KDM inhibitor suggests the effects of alpha-ketoglutarate on the chromatin and on gene expression are relied by members of the KDM5 family. This hypothesis was confirmed by overexpression of KDM5B, which strongly reduced H3K4ME3 levels and markedly increased the cardiomyocyte cell cycle activity when combined with alpha-ketoglutarate. And this is what we can see in the figure 5GH and extended figure 10A through D. KDM5B plays an important role during heart development, and its expression progressively declines during CM maturation. Except we see a significant increase in neonatal 2. We see it in a decrease in adulthood, but we see down, down, then up. Like, so, okay. All right. I'm a bit interested. Like I agree there is a decline, but you can't argue that at neonatal 2. Assuming that this has to be the proper timeline here, they wouldn't, unless they wanted, they wouldn't be that mean to us. So KDM5 knockdown mediated by short interfering RNA effectively antagonize pro-proliferative effects of alpha-KG, fully restoring the reduced H3K4ME3 levels that were lowered by alpha-ketoglutarate treatment, and therefore increasing expression of several key genes associated with cytokine CM cardiomyocyte maturation. All right. So where can we start? I'm busy deciphering this figure right now. I can probably try and help a bit explain it. So the thing is that all of these, one thing to learn when it comes to trying to decipher scientific papers is that you're going to have a lot of language show up, and you just need to kind of gain the general understanding of it. For instance, KDM5 gene family. I actually don't know what the KDM5 gene family does. They briefly mentioned saying that up here, like when you inhibit alpha-ketoglutarate, it will end up pharmacological inhibition of alpha-ketoglutarate dependent effects. It's by the KDM5 inhibitor. So what this is suggesting here is that KDM5, you are using the KDM5 inhibitor to inhibit alpha-ketoglutarate right here. So because of that, they want to look and see if the KDM5 gene family is involved in their pathway. So I can go in and I can start looking up KDM5, trying to figure out exactly what KDM5 does. But for me personally, just knowing that there is a family of genes that will modulate alpha ketoglutarate is enough information for me to continue reading through the paper. So if we want to know more about this, we can. But for now, that should be sufficient. And that's what we end up looking at right here. For instance, we know that there's a family of KDM5s. Here they are. Let's get back over to the figure so we can look. So here's the family of the KDM5s. These 4 are during development. These 4 are during adulthood and during treatments. So when they resulted in doing damage. And here you can say that KDM5 pretty much in these both conditions, it's being upregulated or downregulated. And they ended up focusing on KDM5B. So for me, I was wondering, why are they focusing on KDM5B? And from what I can understand, the reason why they're focusing on KDM5B is largely because it most likely was targeted in an earlier paper. This paper specifically right here. This one done by Zidai, which is mutations in the KDM genes. They result in heart disease. So in this case, they end up showing that in their knockout, so the figure up here, and these figures up here, they would inhibit AKG, alpha ketoglutarate, through KDM5. So they want to finally see, is KDM5 involved? And to do so, they focused on KDM5B because of the fact that it was all looked at previously in the literature. And they show here, I believe here, they were trying to show that KDM5B had a different expression pattern to the other genes. But because A and C and D, like A and C are almost identical to B, and D is the reverse. So I would love to see a later paper published by them where they focus on A and C and see if modulation of them has any impact on this pathway. But yeah, this right here is pretty much them showing off that when you modulate, when you affect KDM5B, which is supposed to be modulating or affecting the levels of AKG or alpha ketoglutarate. Actually, let's hop over to that right over here. Here. So this is pretty much it. This is what I'm talking about. So you have DMSO, you have a decrease in alpha KG. Then when you knock out KDM5B, you have an increase in alpha ketoglutarate. And then when you, this is fold change. This is DMSO. These are the fold changes, the amount. So here you can see a decrease from here to DMSO. Then when you knock it out, you have a significant increase. And then likewise, when you start add in alpha KG and KDM5B, you get a decrease again. So pretty much you can kind of recover from here. So from here to here, you can see there's a similar significant drop between these two, which is what's the important part of this part of the paper. And then likewise, this is all staining to show off what's going on. So yeah, these are all stains, which, yeah, if you don't understand what you're looking at, it looks like a bunch of gobbledygook. But this is one reason why you have to look at the pictures. Or look at the labels down here. So let's see here. This happens to be D and F. D right here. Immunofluorescence analysis of the proliferation marker in the neonatal cardiomyocytes. And this is 4 days after the lentiviral transduction. So this is after damage. DMSO is dimethyl sulfoxide. think of it as think of it as a control. So DMSO, it is pretty much a compound that is used for penetrating cells and tissues. So dimethyl sulfoxide is I didn't know this, it's a byproduct of paper making in Germany. Its usages. So it is a compound that can, all right, so think of it this way. DMSO, you want to get a compound into a cell tissue, but you can't because the cell tissue is too intense. Think of, you want to get a drug through your skin, and our skin is designed to prevent compounds from entering your skin. So how do you get compounds to enter your skin? Well, you would use DMSO. DMSO will break up the it'll make your skin a little bit more transmissible to allow things to be absorbed more easily. Here you go. It absorbs easily into the skin. And as such, DMSO is often or primarily used when you're trying to do stains. Because here we have a cell tissue that has been frozen and it has been put on a slide. So think of you made a big giant ice crystal and put it on a slide, and now you want to stain it. It's kind of hard to get through. It's kind of hard to get through this kind of dried tissue sample that you now have on a piece of glass. So you apply DMSO to help break the bonds up a little bit. And by doing that, you'll break the bonds up, and then you'll allow things to stain it even more and more easily. So that is what DMSO is used for, and that's what its primary use is. It can be used for making it easier to absorb drugs. It can be used to make it easier to stain cell culture. You can use it to. You could use it to. Apparently, you can use it for treating things like cancer and arthritis. So it has a lot of benefits and a lot of uses. I think, let's see here. There should be some other. It is being regulated. It's been approved for treatments. Nice. So yeah, one thing to comment on is right here. So you can see the FDA has approved the use of DMSO for treating cysts, which are pretty much like damage. Cysts are like damage to the skin, or not to tissue. It's tissue damage. Think of hard tissue damage that is very difficult to break down. So the problem is if you overdose on DMSO, you make things too permeable, and then you could technically overdose on the drug of interest. So let's say you want caffeine, you want to get drunk, or you want to get high on caffeine. You haven't had your coffee. So you decide, I'm going to make coffee more effective. I will have a little bit of DMSO with my coffee. You'll absorb a lot more coffee by doing that because DMSO makes it more permeable, makes your cells more permeable. But you might then overdose on caffeine because you ended up having too much coffee enter your bloodstream. So yeah, I hope that kind of explains what DMSO is and how it works and what's its purpose. But yeah, they're using this here as a control, saying, look, we treated our So what they're showing here in their figure is I treated my cells with DMSO right here. I made them more permeable. And because of that, the stains that I'm using, which would be DAPI, which is a nucleus stain, the seractin, which we saw was involved in, this is actin, so it's pretty much a cell tissue marker. And then the Ki67, which is a proliferation marker. Our staining is not because we put too much DMSO on and then everything just got stained with the same way. So that's the whole reason behind this. This is a negative control to show that whatever changes we're seeing here, for instance, we can see we're getting a lot more proliferation in this, I believe this is the knockout. And then here's the one where we added in alpha ketoglutarate. And then here's the combination of the knockout and alpha ketoglutarate. So here they're pretty much, I'm going to have to look. If this KM5B MYC, is this the knockout or an upregulation? I kind of no wait. I don't think this is a knockout. I think this is an upregulated model. Yeah, so this is upregulation model here. So the idea is that these two should theoretically have similar levels, and then we add this in, and then this in, and then to and promote proliferation. And then the idea would then be that this would then we get a lot more proliferation. So yeah, kind of like what we saw here. So yeah, I believe inhibiting this, we end up getting an increase in proliferation, which is what we're seeing. I'm going to have to look a bit more through here. And then here is more stains. Why does this look like... Oh, and this is the different genes. So NAPA-B, MYTH7... oh, wait. Sorry, this is involved here. Oh, yeah. All right, so what is the... So this is for these, and then this is for these. Okay. So this is the knockouts right here. Okay. All right. Yeah, I need to look at the figure because I'm getting a bit lost. I need to know what everything is. So let's see here. So this is D and F. So 10D and F. So where are you? Yeah, 10D and extended 10D and F. So it can be knockdown mediated by short interfering RNA, effectively antagonize pro-proliferative effects of alpha KG, fully restoring the reduced H3KM, that gobbledygook, levels that are lowered by alpha KG treatment. No, go ahead, ask me a question. Again, this is the only way to learn. Ask as many questions as you want. That's the whole point of these things. And it's fun because it helps one also further understand what's going on. To further validate potential targets, KDM5, we carried out CHIP qPCR experiments. We found that KDM5B preferentially binds to different genes with the broad H3KEM3 domains involved in maintaining the mature phenotype of cardiomyocytes such as TINI3, MILK3, MyoCD. By contrast, no enrichment of KDM5B was found on housekeeping genes characterized by narrow peaks such as these guys. MYH7. All right. I believe you're talking about, let's hop over here, this one right here. So these are the different genes. So there's a whole, so G right here is a whole laundry list of different genes. Yeah, so these are pretty much all of the different genes that are involved in a particular pathway. So if I was just actually looking at that right here. So if we look here, we found that KDM5B preferentially binds to genes with this domain that is involved in the mature phenotype of cardiomyocytes. So when myocytes are fully developed, as in when they're in the final, what they're going to be at the very end, then you'll see that there is binding to these particular genes of interest, which would be TIN3, MILK3, MyoCD. They don't mention it, but here you can see MILK3, TN3. Oh, where is MyoCD here? Oh, I don't see it here. But yeah, these are also involved. These apparently are also involved in this pathway, in this pathway right here. In mature, okay, I should say not pathway, but in mature phenotypes of cardiomyocytes. And then they see no enrichment of genes that were involved that are involved in housekeeping. So genes that are involved in keeping the cells happy and healthy. And this would be DUS1, SYNX19, and LSM6. And if you look here, there's DUST1, LIM6, and SNX19. So these are the cell markers, the mature cell markers for cardiomyocytes, and we get upregulation in these. And that here you can see there's no significant difference in the housekeeping genes. And the reason why they're showing this off is to kind of show that the KDM5B and alpha ketoglutarate are involved in the proliferation, but also might have a role in the differentiation of these heart cells. Now, what these cells do exactly, like what does MYH7, what does MYTH7 do? I would have to look that up. I really do not know. Let's see here. So interestingly, they don't actually mention MYF7 in the paper anywhere. So it makes you interested. It kind of makes you curious why they put this in here if they don't even bother mentioning it in the paper. That is kind of what happens with science. It's the sad cyclical cycle of learning. You learn one thing, and so you get an answer to one question, and 3 more questions pop up. For instance, what do if I had to come up with a question, why is this significant? The fact that there's no upregulation of these housekeeping cells. What does that mean? I think they might mention it in the discussions. So I'd have to look at the discussions exactly to see. Yeah, these housekeeping genes that were not affected. Are these the only ones that were affected? For instance, could there be more? Because we're in the cardiomyocytes, which is the heart cells, what do these housekeeping genes do exactly? Because I know in like photoreceptors in the eye, you have a bunch of housekeeping genes, but they're involved in there's a bunch of housekeeping cells and housekeeping genes, but they're all involved in keeping the photoreceptors healthy and happy, preventing them from being overstimulated or preventing a increase in reactive oxygen species and such. So no, it's good. It's good. It's good to have it's good to ask questions. If anyone tells you that don't ask questions, tell them they have poo for brains. Asking questions is the staple of learning and the staple of understanding the world. So if you have a question, ask away. Ask away. Let's see here. Oh. Let's see here. Yeah, let's see. So, yeah, I'm still a bit interested in this. So K67 has proliferation markers. This actin is the stain for the cell bodies, and then DAPI is the let's see here. DAPI is the nucleus stain. So because I study regeneration, but I don't study heart regeneration. And because of that, I don't, there's a lot of things in here that I'm not aware of. So I can kind of glean here and there, but there's going to be a lot of, what does this mean? Oh, I don't know what that means. Oh, what does this mean? So that's kind of what I'm trying to figure out right now. Like, we have DMSO, and then we have KDM5B MYC. I think if I look this up, let me look this up real quick. KDM5B MYC. Aw, come on. What does MYC stand for? Yes, I did say poo-poo brain. I did. I mean, because what else is your brain going to be if you don't ask questions? If you just sit there and accept everything you're told, then your brain is effectively a giant poop. Because you're definitely not adding anything new to it. You're not enriching it, letting it grow. So, all right. But, okay, what is MYC then? Maybe if we look up MYC, it might... blot analysis of MYC targeted... Okay. MYC. Oh, okay. What is... MYC tagged. This is why I don't like acronyms. I hate acronyms. I really do. I know it would make some papers very painful to read, but I would rather have a non-acronym version of a paper than have acronyms, because it's so easy to get lost in the acronyms. Again, I just need to type in I just need to type in AOC, and you'll get so many different things. A MYC tag is a polypeptide protein tag derived from the CMYC gene product that can be added to a protein using recombinant DNA technology. It can be used for affinity chromatography that has been separate recombinant overexpressed. They can be used to separate recombinant overexpressed protein from wild-type protein expressed by the host organism. It can also be used in the isolation of protein complexes with multiple subunits. MYC tag can be used in many different assays that recognize antibodies. So MYC tag is a protein tag that can be used to differentiate between recombinant DNA and, not recombinant DNA. It can be used to separate recombinant overexpressed protein from wild-type protein. So what this pretty much is being used, so if my understanding is correct, MYC means we're looking at normal protein levels or an increase in the amount of protein being expressed. Kind of like when you're doing an RNA expression, or when you're looking at RNA expression, seeing if certain genes are being upregulated. This particular case, we're looking to see if protein is upregulated. So if that's the case, then that means we're tagging KDM5B with this protein. Yeah. Oh, so in this particular case, so that right there is a, all right. Yeah, this is a very fundamental thing. So let me quickly hit this topic. All right. So transcriptome. I think it's called the proteome. Let's see here. Where it oh. Yeah, this works. Perfect. Okay. So this is the basic general flow of when it comes to how cells function. You have DNA at the top, right here. You have DNA. This is where all of the genes are stored. So the gene that regulates coffee, how much you like coffee, that will be in your DNA. Now, based off of how much the RNA is expressed about that particular gene that makes you enjoy coffee, that will be the transcriptome, or the RNA. If you have very low RNA levels, then most likely you're going to not have that gene expressed for loving coffee. So therefore, you won't have as much protein, which kicks in to make you enjoy it. So that's a key. So this is how it goes: DNA to RNA, and then RNA to protein. Now, just because there is an upregulation of RNA does not mean there would be an increase in the amount of protein available. It's possible that the RNA RNA is ribonucleic acids. So DNA is, let's see here, right here, RNA, ribonucleic acid. So RNA is ribonucleic acid. DNA is deoxyribonucleic acid. The main differences between the two is that in RNA, it uses a U instead of a T. So to understand is like, let's see here. Where are the lists of these? The main point is that you have glutamine, arginine, cysteine, and uracil right here. This is uracil. Oh, here it is right here. Oh, hey, sorry, it's guanine. I remembered it though. Guanine, uracil, arginine, and cytosine. And these are the different proteins, different amino acids, I should say. There are different amino acids that make up your DNA. So if you want to make a particular protein, you need to, so think of it this way. Think of this as these are messages. These are codes. If you remember in binary, so in binary, you could have 0001, 0001, something like that. So if I had like 000001 like this, then this code, this code right here pretty much is, the claim is an error code, but pretty much this is a bit, which in programming, this is a bit, which tells, it ends up being converted into something else. So if we ended up doing 1001 in binary, this should equate out to, let's go to the calculator. Yeah, here we go. You're not helping. You're not helping, calculator. Okay. Just want to, all right. So here, 10101010 in binary equates to 170 in math, an actual number. So this right here is a code that the computer understands in order to generate some kind of symbol. So if you ended up putting any type of binary in here and then doing a calculation, for instance, if we change this to 100111 like this, and then we tried to calculate, it became out to be 143. And then we could, let's see here, sorry. And then we could change this, maybe, we'll just do 1000 this time. 8, see? So this is what that does. The same thing is true for biology. However, instead of using zeros and ones, all right, so instead of using zeros and ones, nature decided we're going to use 4 amino acids, a G, a U, an A, and a C for the RNA, and for DNA, it will be a C, a G, a T, a C, and an A. And what you do is you combine these together in these 3-part codes to make different, make different proteins. Those are called codons. So codon. So this is called a codon right here. Right here. There we go. Nice. Okay, so here we go. Okay, not so nice. And also not so nice. Come on, we want to... Where's the nice little example? Maybe if I look over here. Looking for a nice little example that doesn't make people want to vomit. I guess this works. Codon. And it's forbidden. Ah, here we go. Perfect. This one right here. Here we go. Perfect. Okay. So here we go. Here we go. These are the different codons. You can see GCA is a codon. CAC is a codon. TAC is a codon. GTG. This is all DNA right here. And then here you have RNA right here, which is CGU, GUG, AUG, CAG. So here you can kind of see the different, this is how the codons work. And they all promote a different code. Sorry I'm bouncing around all over the place, but this is the best way to explain it. Here is all of the different codes that are available. Here you go. So here are all of the codes available and how they all go to where, how they all direct to a particular protein. So for instance, the TGA, what you would find in the DNA, a TGA, it'll be transcribed to RNA as AUT, and then AUT will go to the proteins and then say, stop making proteins. You're a stop codon. You stop making proteins. So that's pretty much how all of this works. You go from DNA, let's see here. So you go from DNA to RNA. You go from DNA right here to RNA, and then to protein. And it's all in the vein of making it uses these codes, and it's all in the vein of making different protein structures. And to kind of explain generally, have you ever, oh, well, they're all called ribonucleic acid. Oh yeah, so a way to explain it. So in regards to why they're called ribonucleic acids, it is actually because of the fact we're not thinking of acidic base. Well, we are thinking acidic basic here. But the main thing is that it means that they have free electrons. It's all about we're going to get into a bunch of physics and chemistry here if we go that deep. How much do you want to? We're now diving into the world of physics and chemistry. The main thing is that they have the way that they bond. The way that they're bond and what's so. Yeah, we're going very off topic of the paper. So let's quickly go to. So let's see here. We're looking for. Let's just type in why is DNA an acid? Oh, whoops. DNA is an acid. There's a nice little stack exchange called biology, which explains these things. See, DNA is not made up of nucleotide bases, but of nucleotides. These consist of sugar bound to one of the 4 nucleobases, adenine, cysteine, guanine, and thymine, or uracil in RNA, and a phosphate group. When these bases are bound to a sugar, the deoxyribose for DNA form the nucleosides, adenosine, cytosine, guanosine, and thymidine, respectively. When the nucleosides are bound to one or more phosphate groups, they become nucleotides. The generally structure of a nucleotide is this. Finally, nucleotides are joined together through their phosphate groups that form phosphodiester bonds between the 3rd and 4th carbon atoms of adjacent sugars and rings. This polymer is what we know as DNA. Right here. So, after this long introduction, the acidity of the DNA is caused by the presence of the phosphate groups, which are themselves acidic. You can see this by comparing the DNA structure to that of phosphoric acid. So, there you go, phosphoric acid right here. Actually, that looks very much like interesting. If you compare the structures of phosphoric acid and the short strand of DNA, you'll see that the latter 2 protein of phosphoric acid are replaced by carbon atoms either in or attached to the 5-member ring. In chemical terms, such a group is called the phosphate diester. The phosphate diester. The remaining proteins, the remaining proton is now quite acidic and is relatively easily lost, thereby giving DNA its acidic character. Indeed, under natural conditions, DNA is deprotonated at this site, and the oxygen atom bears a negative charge. Despite the fact that DNA does contain many basic groups, their basic properties are masked somewhat because of the fact that they hydrogen bond with each other to form base pairs. Hence, it's the acidic part of the molecule that dominates, and that is why we know DNA is an acid. Hopefully that explains it. It's again, it is. So phosphate is it's a chemical. If you look here, phosphoric acid, there's the phosphate right there, surrounded by oxygens and hydrogens. And there's nitrogen right there. There's the N, that's a nitrogen. I think there should be, you don't see them here, but you see these sites right here, they're carbons. Because organic chemistry is like 99% of organic chemistry is carbon-based. That's what organic means in organic chemistry, is carbon. So they just ignore putting carbon down. So carbon, carbon, carbon. These are double bonds to carbons. There's a nitrogen. There's a nitrogen bound to a hydrogen. Yeah, it's pretty much chemistry. Yeah. So the reason why it's DNA is considered an acid, if we go back to the name, it's deoxyribonucleic acid. I wanted to kind of. Oh, actually, wait. I think if we go. All right, it's DNA, deoxyribonucleic acid. The deoxyribo is for the nucleotides, and the acid part is talking about the phosphor bonds. Yeah, carbon, carbon, carbon. Exactly, exactly. Carbon, 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 carbon. But yeah, so with that in mind, sorry, I'm busy quickly looking through here to see if I can find the. All right. So with that in mind, what you have is you have DNA, which will form RNA, and then RNA will be translated in these protein structures right here, which will make these different proteins. So what happens is you have the anticodon with the amino acid. They'll come in. You don't need to, if you want to get more into the chemistry of the things or the physics of things, you can learn about this structure. But because we don't want to go that deep, the main thing you just need to know is that the amino acid, you take the DNA, which makes RNA. This RNA then is translated to form these different proteins. And based off of the codons that are used, based off of these different codons, you will make different nucleotides. That's the name of the A, T, G, and C. That's the name of the A, T, G, and C. It's the grouping that they call it. But yeah, from there you'll then make these different proteins, and then the proteins will have a particular structure to them. So let's see here. Let me pick an easy one. Sickle cell. Have you heard of sickle cell anemia? Sickle cell. Come on, internet. Don't die on me. It's dying on me. Okay, the internet's it's having trouble. But sickle cell anemia is caused when there is misfolding of the hemoglobin in blood. And because of that misfolding, and the misfolding is caused by one codon, one mutation in the one area in the DNA, which makes the red blood cells. So you have a mutation in one of the codons, which results in one of the proteins being wrong, which results in malformation of the red blood cells, which results in a sickle cell, which is what causes sickle cell anemia. So it's very important for the codons to be correct and for the DNA to be correct. And the reason why we're trying to figure this out is because what's going on here in this paper, or we're going to loop back to the paper, what's going on here is the only way to treat sickle cell is to go in and physically change the stem cells in the bone marrow, repair or change the DNA in those stem cells, which are generating the blood cells. And the only way to do that is through like a viral vector, which is extremely dangerous, or through other means. Well, yeah, sickle cell is a genetic disorder. It's a genetic disorder which is caused by a mutation in the DNA, which results in a mutation in the hemoglobin in red blood cells. So it's not a genetic disorder. Or, I mean, sorry, it's not you can't catch it. It's not a bacterial infection or a viral infection. Yeah, disorder. It means it's not working properly. For instance, there's a brain disorder when I forget words, or a speech disorder when I fumble my words and I start saying things inappropriately, like messing up word structure. Yeah, disorder. It just means it's not working properly, or it's not working as it should normally work. But I think I figured out what's going on here. Because they want to decrease protein levels, they are using MYC to bind to the protein, inactivating it. So here they are deactivating KDM5B. So DMSO deactivated KMD5B, alpha ketoglutarate, and then deactivated alpha ketoglutarate with deactivated KDM5 with alpha ketoglutarate, and then this is why we see this increase in the amount of cells that are proliferating, because we know that alpha KG is supposed to be involved in proliferation, and if you knock out its inhibitor, you get an even greater proliferation. And this is gene expression, which is what was confusing me, because I was trying to compare these two, and yeah, they're different. So sickle cell trait is a distinction of blood cells. Yes, exactly. You're right. It is a distinction. Sickle cell is purely a malformation of the hemoglobin in someone's blood. And it is surprisingly defined by a single protein being a single protein, a mutation in a single protein. If this would work, we could see. But sadly, yeah, it's not working. Wait, here we go. Here we go, perfect. This is exactly what I'm talking about. Here it is right here. Oh, this is very nice. Okay, so here we go. Normal hemoglobin. It has a glutamate in position 6. This is what the structure looks like. And this is the resulting protein structure. And this is how the red blood cell looks in the end. So yeah, that right there is a normal red blood cell. Sickle cell. You have a valine in position 6 as opposed to the glutamine. This results in this structure right here. You can see there's a change from red to blue. If you'll have to understand the chemical structures, because the glutamate has 3 binding sites while valine only has 2. And you might think, well, that shouldn't be a huge amount of difference. But because of the fact that there's a difference in binding sites, you result in a malformation in the protein. So you end up getting this going on, which results in sickle cell like this. And that's pretty much that right there is the definition of sickle cell. All right, there we go. Anyway, back to this. We're almost done with the paper. Let's get done with this. So yeah, so here this is showing this off, and then likewise, here is this is the KD. So I believe what this is referring to is they're going to upregulate KMD5B, and by doing so, we get a decrease in the amount of proliferation. So this is just showing the connection between alpha ketoglutarate, KMD5B, and the pathway, and then finally, in the last bit of the figure here at the bottom, this is showing off the potential pathway. So this is what they believe the pathway is. So hey, the TCA cycle. This is the energy cycle right here. So we have CPT1. There we go. We have folic acid going to CPT1B. Then we get acetyl-CoA, which is an amino acid. We then go to citrate, isocitrate, and then we break down to alpha ketoglutarate. We then go to succinyl-CoA, and then we go to oxyacetylate, there again. All right, we go to this one, and then we repeat. So this is the Krebs cycle. And then when we knock out CPT1B right here, we end up having glucose and pyruvate going in, which will then PDH increase, which will then go through the cycle. But as such, we then have an increase in production of citrates, so citrate will come out. And then likewise, we'll have an increase in alpha ketoglutarate, which will come out. And then these will then leave and then enter the nucleus right here. In which case you see it doesn't happen in normal conditions, but in their CPT1B knockout, you'll have this happen, which will then result in binding to KDM5, which is found here on the DNA, which will then result in decreases in these genes here. So this is the perspective pathway that they're arguing for regeneration. Ooh, very interesting, very interesting. It's actually a bit different from the pathways that I usually look at when it comes to when I'm looking at regeneration in zebrafish and such. Because in the regenerative models that we usually look at, let me show you what those usually look like. So regeneration pathway zebrafish. So when we're usually looking at this pathway, maybe if I type in some genes, STAT3, WNT, SOX3A. Let's just get some genes in there. There it is. See, this is what I'm usually thinking of when it comes to regeneration. And this involves a completely separate pathway. If you notice, you have STAT pathway, you have the WNT pathway. The main point you need to know is these are all involved in proliferation, regeneration, and development. We can go into this later. But the main point is, as you can see, these are all different genes that are involved in other pathways. It's quite intriguing to see that they have a paper which is looking at the mitochondria and its and potentially its role in regeneration through the Krebs cycle. So if I was continuing research and doing more research, I would actually be looking and seeing alpha ketoglutarate. Is there any potential pathway here that might involve alpha ketoglutarate in here? And what about the CPT5B? It's a binding site on the DNA. I know of beta-catenin, and well, here's beta-catenin, which is part of the WNT pathway, but like, so it's quite curious, quite curious. Very interesting, very interesting. And this is all about promoting proliferation too. So I was thinking about, because when they were talking about regeneration in the heart, one of the first things I thought was they might be modulating the STAT pathway, because STAT pathway is involved in proliferation. Or they might be regulating the NF-kappa B pathway, which is involved, it's also involved in regeneration. WNT pathway is involved in development. So this is a development pathway. So it would mostly be involved in like differentiation and de-differentiation. So I was expecting to see a lot of these things, but it's actually a bit surprising that here we have heart regeneration going on and not the usual suspects. Not the usual suspects at all. That's very intriguing to me. It's possible that I've been out of the loop for so long that this might have been found a while ago, but for that case, I would have to go back and start looking at even more older papers. And then this is the methods and such. So the methods and the such. So, but yeah, this is a very. I can see why it was published in Nature. Oh yeah, I can definitely see why it was published in Nature. It's a very interesting paper. It has and it's all about oxidation, fatty acid oxidation. Kind of makes me wonder about fatty acid oxidation again in just normal regeneration. So let's go with stat 3. Oh, when was this published? 2018. This is in the cell metabolism. So the STAT pathway, regulated fatty acid beta oxidation, is critical for breast cancer stem cell self-renewal and chemo resistance. Interesting. All right, so 2018. So that is still pretty. I mean, it's still pretty recent, actually. It's 6 years old, but all right. Interesting to know. So yeah, apparently this is much further down in the pathway. Yeah, maybe. Anyway, it's. And then after the paper, I want to talk about sickle cell since I already understood. Yeah. As you go through papers more and more frequently, you slowly start picking up on these, and you'll eventually go, oh, I know this, I know this. So let's see here. It takes time, but once you figure it out, then once you start, because this is a lot of knowledge. I mean, there's this one figure that since we're talking about the Krebs cycle, because if all right, let me show you. Let me put this in perspective, because you have to learn to hyper focus on details and not just start asking questions down a rabbit hole, because it is science is literally a never-ending rabbit hole. You might go down one path and you might never make it back out because there's just too much information. So, sorry, here. This right here, Krebs cycle. It looks nice and clean, doesn't it? Here you have glucose, pyruvate. You can see it sometimes enters, not so much, but then you have fatty acids entering through CPT1B, which then goes through the cycle, and then you get energy production, and then that's all good. Do you know what the actual Krebs cycle looks like? I still remember this back in undergraduate because I had a professor who posted the entirety of the Krebs cycle that we currently understood. So, full Krebs cycle pathway. I wonder if I can still find that picture. And of course, it's not going to work. I wonder why this internet is acting so slow right now. Maybe it might be DuckDuckGo. Let's try Google. Let's see, Krebs cycle pathway. There we go. So here we go. So this is actually a bit more detail about the Krebs cycle, as you can see right here. So you can see here, this is a little bit more detailed. So actually, it's not just the ones that we saw. For instance, we saw citrate, we saw alpha ketoglutarate, succinyl-CoA, and then we saw it also talked about oxaloacetate. But actually, there's cis-acyanate, D-isotrate, isocitrate. There is succinate, fumarate, malate, and then here you have water entering. You have other like NAD, NADH. Here we have succinyl dehydrogenase. Like we have a whole, like this is a complex pathway. And this is, again, let's zoom out even further. Let's zoom out even further. Instead of just the Krebs cycle, let's do the energy pathway. I wonder if that will work. The full human energy pathway. No, we're getting a bunch of BS. Let's see here. The full energy pathway and cycle. Oh, I sadly I don't think I think they're not going to show me this. I think they're not going to show me this. I think what we're going to have to do is just keep slowly building up, because now here we have things going on outside, like glycolysis, lipid synthesis. We have aerobolic glycolysis. We have lipid metabolism. We have glutaminosynthesis. And these are all different pathways that all expand out into another area. For instance, like here, yeah, these all expand out to a different pathways here. And I wish that I could find it, because the poster that he showed me back in undergrad had over 1,000 genes on it, and that was just we currently know. So let's see here. Oh, I know. We will go with the full metabolism pathway. There it is, right here. Can I see the picture, please? Oh, sorry. Oh, it's not going to show. Let me see it, is it? Maybe if we go back real quick and just load it up. Just load up the image. Ah, this is good. No, it's not good enough. Come on, let me show off the picture. We want to have fun. I just want to see this one picture that's maybe this will let us see it? Come on, let me see. Let me see. No, it's not letting me see. Oh, come on. Why won't you let me see? It's such a basic pathway. Like, finally, here we go. I'm bored. All right, here it is right here. Just know that every single dot here is a gene. So this is the current understanding of the metabolic pathway. Let's see here. So yeah, you can see it's very complicated. It's very, very complicated. But yeah, that's pretty much all. That's pretty much it. We're done with the paper. We're done with the paper. So there's a lot of different avenues to start testing on this paper. By the way, there we go. That was fun. We'll go through another paper next time. I was actually hoping to have gone through this a bit more quickly, but it's maybe we should pick a smaller paper next time. Maybe something not from Nature. But yeah, it's very interesting. Very interesting. Oh, I hope they'll be able to make drugs for this pretty soon. Your micro HDHD has not been entertained. Ah, funny. All right. Either way, I'm gonna hop off now. We'll do more paper crunching next time. Either way. See you later, Tapper. I'll hop off now. Laters. Laters.